Global challenges such as climate change and micropollutants have compelled the European Union to embrace sustainable bio-based production. In this context, the EU-funded project PROMICON aims to harness highly productive microbial communities for the clean production of sustainable products such as hydrogen gas, lipids, and chemicals. Microbial communities have powerful traits like stability and functional robustness, and they can perform complex tasks. Omics profiling acts as a language to understand and communicate with these communities. However, when multiple microbes are speaking at once, it's challenging to know who is saying what. To address this, PROMICON is developing an OMICS platform to better translate microbes' language and differentiate the voices within the consortium. This improved conversation with each individual in a microbial community would allow for more precise control and optimization of microbial functions to enhance production. PROMICON researchers combined metabolomics, proteomics, and stale isotope labeling technology. Their work consists of two main components: establishing a microbial consortium to produce hydrogen, and developing an omics platform to understand the mechanisms of this consortium better.
So here is our lab of reactors. You can see we have different units of the reactors, and this system named DASGIP system. It's an automatical controlling reactor system which could help us to control different pH, temperatures, and also light conditions. Also, we have different sensors like the dissolved oxygen, pH, and all of this could help us to automatically regulate the reactors and regulate what is happening inside. Let's take a further step to here and look into the real running reactors here. Actually, now we are co-culturing two strains together here. One is the cyanobacterium, the Synechocystis, and another is the non-sulfur purple bacterium, the Rhodopseudomonas palustris. And the first, the cyanobacterium, they could actually fix carbon dioxide from the air and convert them into different organic acids. And once they release this organic acid into the environment, the other strain, the non-sulfur bacterium, they would uptake these organic carbon sources and use them to produce hydrogen. And hydrogen is the final product of what we want, because that's a kind of the clean and renewable energy, and that could also help us to address the problems of energy shortage now we are facing. Actually, before the detection and the quantification of any omics data, we need to prepare the samples. We get the biomass from the other lab, and then we were using these machines, lyophilizations or centrifuge ones, and also depth centrifuges, using all of these machines to pre-treat the samples, preparing the samples, and for the later omics study, and that will including the metabolomics and also the proteomics data. For example, we have the GCMS, the gas chromatograph mass spectrometry, and using this one we can identify and quantify some of the compounds, for example the lipids and the fatty acids, and also some of the sugars compound and so on. And this is the first part we are going to detect. That's the HPLC, the liquid chromatograph, and also coupled with this ORBITRAP, the MS system. And using this system, actually we can also quantify, for example, the amino acid groups or some of the CoA compounds from the metabolites, and then we can say what they are doing, and we can get some information from there. Except for this one, we also have the ICMS. It's also this machine coupled with the ORBITRAP system. That's the mass spectrometry. That's a very high resolution mass spectrometry, which enable us to detect the different metabolomics informations in a very broad way. And we can also even couple this one with the stable isotope labeling part, so that we can understand more accurately what is happening and what we want to get from the microbials. when we combine all of these machines together, we can get an overview about the consortium, how they are doing and what they are talking to us, what information they want to give to us. And later, using all of these informations, we can better regulate the system. We can better regulate the efficiency and better let them to grow better. And that's our final goals of using all of these machines to understand them better. The next step, after we receiving the raw data from the lab, and then we are going to start to analyze the data. Why we want to do this? That is because it's like a language translation process, which we want to translate their information and their language into the one which we can understand better. Actually, we need to do a lot of different things to treat the raw data. For example, we using the R Studio, the different R code to treat with the metabolomics and the proteomics data. And then we also use the different softwares, for example, this compound discoverer or the proteome discoverer to treat different data set. And with all of these analysis, and we can grab different informations. And we also categorize these informations into different ways and then summarize them again. And then later we can draw some conclusions. And this information really help us to understand what they are doing and the mechanisms. And then as a return, we can improve our efficiency, improve our experiment according to this result. And that's really helpful and also quite meaningful for both research and the applications. Still, we have some further improvement space, what we are going to do in the next step. For example, we are also going to combine our OMICS platform with the 13C isotope labeling this kind of technology, and so that it could give us more accurate result and the informations. And that's the overall goals and the overview of our project.